Routine lab tests are not reliable for diagnosing long COVID, according to a new study. The research found that no clinical lab values could serve as biomarkers, highlighting the need to focus on symptoms for diagnosis. A new study found that most routine laboratory tests are not reliable for diagnosing long COVID, also known as post-acute sequelae of SARS-CoV-2 infection (PAS). The study, published in Annals of Internal Medicine, found no reliable biomarker among 25 routine clinical laboratory values for prior infection, PAS, or specific types of PAS clusters. This suggests none of these routine labs can serve as a clinically useful biomarker of PAS. This study shows patients can have severe long COVID with normal lab results. This suggests doctors should not focus on the results of blood panels to diagnose long COVID, but should focus more on symptoms and ways to help patients get relief by treating their symptoms. 7% of all adults in the US, nearly 18 million people, currently have long COVID, according to the Agency for Healthcare Research and Quality's Medical Expenditure Panel Survey. For researchers, the challenge is to discover biomarkers that can help us quickly and accurately diagnose long COVID to ensure people struggling with this disease receive the most appropriate care as soon as possible. Long COVID symptoms can prevent someone from returning to work or school and may even make everyday tasks a burden, so the ability for rapid diagnosis is key. To investigate clinical laboratory markers of SARS-CoV-2 and PAS, the researchers examined data from nearly 10,000 adults with and without SARS-CoV-2 infection. The researchers recruited from over 80 enrolling sites across 33 US states, plus Washington, D.C. and Puerto Rico, making it one of the largest and most diverse studies of its kind. The study compared results in several ways between participants with and without prior SARS-CoV-2 infection at 6 months after infection, between participants with and without PAS, and between participants with each of the 4 most common PAS symptom phenotypes and those unlikely to have PAS. Researchers found participants with prior SARS-CoV-2 infection showed modest increases in HbA1c, a marker of long-term blood sugar levels, and urinary albumin to creatinine ratio (UACR), along with small decreases in platelet counts. While these differences are statistically significant, these associations are generally small and not reliable enough to serve as diagnostic biomarkers for PAS. The researchers suggest this data shows the complexity of PAS as a condition that may involve multiple physiological pathways beyond simple laboratory markers, such as those for inflammation, anemia, or other markers. Long COVID has been very elusive. Numerous possible symptoms, no definite cause, and no clear treatment. We hear from patients that their concerns are dismissed by providers because their lab tests are normal. In this study, even the exhaustive list of routine blood tests could not help in making a PAS diagnosis. This is an important observation in past research, as prior smaller studies showed inconsistent abnormalities in some blood tests. Until a reliable biomarker is found, the best diagnostic modality for PAS remains the old-fashioned history-taking and clinical assessment. The researchers also note that it's still important to do routine laboratory tests to rule out other conditions during the process of diagnosing PAS. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.